on behalf of the UNC Institute of Pharmacogenomics and Individualized Therapy, I'd like to welcome you here. The Institute was founded in May of 2007 with resources from around the university. School of Pharmacy was the main contributor, School of Medicine, School of Public Health were also major contributors, and then of course the Lineberger Cancer Center and the Center for Genome Sciences had a major role in this development. And one of the things that we were doing is developing interdisciplinary approaches to tailoring therapy and trying to really push the application of individualized medical practice. And as part of that, we were emphasizing development of the science and of the processes of application. But we realized that a lot of work had been done for us by people who currently didn't have a real avenue of recognition. And so we set up an award, a selection of awards, 3 in particular, where we each year recognize individuals that have stimulated the field and really have plowed the way for us so that our lives have been made easier. And these 3 areas are in patient service, public service, and in clinical service. And today we're going to be giving out the award for clinical service. This is an award that annually honors a person who has made significant direct contributions to the advancement of individualized therapy in clinical practice. And the awardee for 2009 is Dr. Richard Schilsky. Dr. Schilsky is a professor of medicine at the University of Chicago and the chief of the section of hematology and oncology at that university. Received his MD degree from the University of Chicago Pritzker School of Medicine. I won't say which year. He was followed by training at UT Southwestern and at the National Cancer Institute. He's a hematologist-oncologist by training and a clinical pharmacologist by expertise and training, and has made significant contributions to the area of both drug development and clinical trials, including a lot of work on the multi-investigator NIH types of studies. Rich has been the chairman of the NCI Clinical Trials Cooperative Group, Cancer and Leukemia Group B, CALGB, since 1995, and is really leading a complex organization of both academic and community oncologists that are driving drug development forward, mainly in the areas of phase 2 and phase 3 drug development. He has served on a number of peer review and advisory committees at both at the NCI and otherwise. He's a former chair of the Oncology Drug Advisory Committee at the Food and Drug Administration, and currently serves as chair of the NCI Board of Scientific Advisors and as a member of the Clinical and Translational Research Advisory Committee. He's also served on as a member of the Board of Directors of the American Society of Clinical Oncology and was the ASCO president for 2008-2009. And so he's just still recovering from that particular task. Dr. Schilsky was selected as the recipient of this, the 2009 IPIT Clinical Service Award because of the dramatic and revolutionary improvements he has caused in the area of individualized cancer therapy. Dr. Schilsky has been directly responsible for shifting the discussion from should we individualize therapy to how do we pursue individualized cancer therapy. And that change has caused a lot of practical activity to happen. Just within CALGB alone, we now have integrated pharmacogenetic sampling into all large clinical trials. Over 40,000 patients worth of pharmacogenetics being done across that cooperative group at a time when just 5 years ago there was none anywhere in the nation. So it's really dramatic changes that have happened. And so his advocacy for practicing medicine based on individualized patients rather than a generic approach has been really helped move this field forward. So Rich, if you could step forward here. Maybe we can go out here for the camera's sake. I'd like to present to you the 2009 IPIT Clinical Service Award. Okay. Great, thank you. And now I'd like to also welcome you to come and go ahead and give your presentation on personalizing cancer care, research policy and practice. Oh, and sorry, Dr. Goldberg has something else for you, it sounds like.
Before you educate us, not to be outdone by Clara at the CLGB meeting, who gave Rich an Ohio State football. We have a Butch Davis, the football coach, signed football
signed.
jersey.
Okay.
So while you're throwing the Ohio State football around with your young division members, you can wear this jersey.
Fantastic.
Wear it in good health.
Well, thank you so much. Oh, this is fantastic. All right. Great. Thank you, Rich. And let me
And
turn
you don't realize what an occasion this is because we've never seen Howard in a suit before.
I was remarking on that earlier today when I was overwhelmed by seeing him in a suit and tie. Well, thank you all for this wonderful award and great honor. It's been a good year for me this year with having been ASCO president at the meeting in June, and I'm in the process of trying to retire as the group chair of CLGB, and we just had a wonderful party last weekend in Phoenix at our meeting. And so this is sort of capping off a terrific week for me. Howard said many nice things, but I must say that I think many of you here are really at the forefront of doing what we all think we need to do in improving cancer care for patients, which is personalizing cancer care. And so I think much of what I'll discuss with you today is probably well known to some of you, maybe less well known to others of you, but I have a few themes I'm going to try to develop in the time available. And I have a fair amount of information. I may sort of need to zip through some of it in order to get to the punchline, but here we go. So why is this an area that we want to pursue at this point in time? Well, I think it's pretty clear that we have a better understanding of tumor biology than we've ever had. Not as good as we need to have or would like to have, but probably more insight into cancer biology than ever before. Importantly, I think we also have technology that's available now that allows us to assess variation in both germline DNA and somatic variations in tumor DNA. And so that has provided us with a new opportunity to match the treatment to the tumor, to select the optimal dose for an individual patient, to optimize the entire drug development process and hopefully make it faster and more efficient, and ultimately to reduce the cost of cancer care by better matching therapies that are likely to work to patients who are likely to benefit from them, and not giving costly and toxic treatments to people who won't benefit. Oncologists, of course, have known for decades that the experience of each cancer patient is unique. All experienced clinical oncologists recognize that each person's presentation can be variable. Certainly, each person's prognosis is variable. Patients' response to treatment and tolerance of treatment is highly variable from one person to the next. And increasingly, we're beginning to recognize that even everybody's survivorship experience is variable. And as you know, we have many more cancer survivors in this country than ever before. And estimates are we have about 12 million of them in the United States. And every single person who's a cancer survivor has a unique risk of recurrence, a unique risk of developing a second malignancy, a unique toxicity profile that needs to be managed as they go forward. And the same is true, of course, with respect to end-of-life care for patients. What we've learned in recent years, of course, is that each tumor is unique. Every kind of cancer that we study is really a mix of many, many subtypes of cancer. These are just illustrations for diffuse large B-cell lymphoma and breast cancer, showing some of the subtypes of those tumors. And I would suggest to you that the fundamental core of the cancer problem is the plasticity of the cancer genome and its ability to generate enormous heterogeneity. And that is what creates both a challenge to us and also the necessity for us to personalize therapy, because every tumor is different in every patient. And to optimize treatment, we really have to understand that. Increasingly, of course, we also understand that drug effects are variable, and this is really part of the core mission of IPIT, of course, is to understand these things better. So we know that all patients respond to treatment differently. We've had a tendency in oncology to derive drug regimens based on experience in a broad population of patients, and then to administer the same dose and schedule to everybody who walks in the door with a given diagnosis. And that is far afield from other therapeutic areas in medicine, where we do a lot more therapeutic drug monitoring and make attempts to individualize treatment. And increasingly, we recognize that much of the variability is due to genetic factors that may impact either drug transport into the cell, a drug metabolism in the cell, binding of the drug to its target, or in fact, the nature of the target itself. So let me give you a few examples. And I have a sort of a hypothesis or contention that I want to lay out for you, which is that although we're learning a lot about how to individualize treatment based on biomarkers, I think that where we are right now with biomarkers is that single gene biomarkers in particular are going to be much more useful to us in figuring out who should not receive a particular therapy than who should receive a particular therapy. And I think, you know, some of our best biomarkers that are in clinical use are well suited specifically to that purpose. This is a study that we did in CLGB a number of years ago, a study called 9344. This was an adjuvant study in patients with node-positive breast cancer who were randomized to get standard AC adjuvant chemotherapy with or without the drug Taxol. And this is the typical kind of adjuvant chemotherapy curve that one often sees. You know, it's clear that the patients who got Taxol did better. It's also clear that the incremental benefit is small. And, you know, surely it must be obvious that it's unlikely that everybody in this study benefited a little bit, and what is more likely is that some patients benefited a lot and some patients didn't benefit at all, and our job is to figure out who is who. Taxol is a toxic drug, and we would prefer not to give it to everyone. So we did some biomarker studies. One of the things that we've tried to do in CLGB over a long time now is to be sure that we have tumor specimens collected whenever possible on patients who are on our study, so that we can begin to look at molecular features that may correlate with outcomes. And what we found, this is a paper that was published a couple of years ago, is that there's a group of patients who turn out to be the majority of the patients in this study who are characterized by having HER2-negative, ER-positive breast cancer who clearly have no benefit from the addition of Taxol to their adjuvant chemotherapy. And then there's another group of patients who are HER2-positive, ER-negative, who have quite substantial benefit and in fact derive most of the benefit from this treatment. So going forward, I think one could reasonably hypothesize that this is the group that should get adjuvant Taxol, and this is the group that should have Taxol withheld. And so we now have these biomarkers of a group of patients in whom adjuvant Taxol appears not to be beneficial. The same is true, you know, as one begins to move into more sophisticated molecular profiling, and this is the Oncotype DX test that I'm sure many of you are familiar with. This is a 21-gene profile. The genes are shown on this slide, and they include some of the genes that we know to be important in breast cancer and other genes like Ki67 that measure proliferation, some genes that measure invasion, and then there's a group of reference genes. You know, the data for the Oncotype DX were also developed from analysis of specimens collected in a cooperative group study and some studies done by the NSABP. And you know, what they found was that they could identify a group of women with node-negative, ER-positive breast cancer who were receiving adjuvant tamoxifen who had a low, intermediate, or high risk of recurrence that could be defined by the gene expression signature of that 21-gene profile. And that then allowed for the development of a recurrence score and the separation of these patients into low, intermediate, and high-risk groups. And this is now a commercially available test. Most doctors who treat breast cancer patients order it fairly routinely in the appropriate patient population, and it's used as a guide in helping them make clinical decisions. Now, the risk score was originally developed to predict the risk of recurrence in patients who are receiving tamoxifen, but it turns out to potentially also be beneficial in predicting the risk or predicting the utility of adding chemotherapy to tamoxifen. So this is another study from the NSABP where they looked at the benefit of adding adjuvant chemotherapy to tamoxifen based on the Oncotype DX risk score. So again, here's the results for the overall study population. And again, it's the same sort of curve that I showed you for the other breast cancer study. A definite improvement for the study population overall for the addition of chemotherapy to tamoxifen, but a small overall difference. And then if you apply the 21 gene signature and parse out low, intermediate, and high-risk patients, you can see that all of the benefit of the adjuvant chemotherapy when added to tamoxifen is in the high-risk group. And there's essentially no benefit in the low-risk groups. So these data have led to an ongoing prospective randomized clinical trial known as the TAILOR-X trial, which is attempting to really determine the clinical utility of this Oncotype DX assay to identify patients who can forego adjuvant chemotherapy if they have node-negative, ER-positive tumors. So the study is shown here. The patients who fit that description have the assay done on their tumor block. Patients who have a low risk of recurrence are assigned to receive endocrine therapy. Patients who have a high risk of recurrence are assigned to receive chemotherapy with their endocrine therapy. But patients in this intermediate group, where it's unclear whether there's really benefit associated with adjuvant chemotherapy, they are randomized to get endocrine therapy or endocrine therapy plus chemotherapy in a study which is a so-called non-inferiority design. So the study is designed to show that giving only endocrine therapy is not inferior to giving chemotherapy with endocrine therapy. This is a large study. It's going to take about 11,000 patients to answer this question because of the non-inferiority design. But it has the potential to be able to use this test to safely omit adjuvant chemotherapy in this group of women. There's a similar trial that's going on in Europe known as the MINDAC study, which uses a different expression profile, the so-called 70-gene MammaPrint assay, another commercially available assay now in the US. And I won't go through this in great detail because it's a much more complex study design, but it's essentially designed to answer the same question. That is whether women who are determined to be at intermediate risk of recurrence benefit from the addition of chemotherapy to endocrine therapy. And if they don't, then hopefully to be able to omit the adjuvant chemotherapy. And we're seeing the same theme play out time and time again now across a variety of different tumor types. This is another study, the so-called ILT study, an adjuvant study of cisplatin-based chemotherapy in patients with resected non-small cell lung cancer. So again, here are the overall study results for overall survival. Again, a statistically significant but small incremental benefit in terms of survival in favor of giving adjuvant chemotherapy. So, same question, can we cull out a population of patients who are the ones who really seem to benefit from the adjuvant chemo and withhold it from the others? So in this case, the investigators looked at expression of the ERCC1 gene in these tumor blocks. ERCC1 is a DNA repair protein that seems to be of importance in repairing cisplatin-induced damage. And you can see again that patients who have ERCC1 negative tumors, now there seems to be more obvious separation of the curves, and there's a more clear-cut benefit of adjuvant chemotherapy, presumably because they are not able to repair the damage from the cisplatin, and it's more effective. Patients who have ERCC1 positive tumors, in fact, not only is there no benefit from adjuvant chemotherapy, but those patients may even have worse outcomes. This is a test now that is being studied more intensively, both in non-small cell lung cancer and in colon cancer, where oxaliplatin-based chemotherapy is so important. Probably the best example of using a biomarker to predict how not to treat patients is the whole story that's evolved in the last couple of years about the impact of KRAS mutation status on the use of the anti-EGFR directed monoclonal antibodies in colorectal cancer. So as I'm sure you know, there are two antibodies that are on the market now, cetuximab and panitumumab, that are important components of treatment for patients with advanced colorectal cancer. They can be used as single agents. They're often used in combination with chemotherapy. And like so many treatments that we use in treating cancer patients, some patients are helped and many patients are not helped. The question again is, can we figure this out? Well, so the data are showing very clearly now that if one looks at patients who have mutation of the KRAS gene in their colon cancer, clearly do not benefit from the addition of cetuximab or treatment with cetuximab. This happens to be a study of, this is actually with the enzyme, with the antibody panitumumab, comparing panitumumab to patients with best supportive care. So single agent study, KRAS mutated, no benefit of the panitumumab compared to best supportive care. KRAS wild type patients, considerable benefit for this treatment approach. The same is true for the more commonly used drug cetuximab. Again, here a randomized study looking at cetuximab compared to best supportive care in colorectal cancer patients with either mutated KRAS in the tumor, where clearly there's no benefit, or wild type KRAS, where clearly there is a much more substantial benefit from this treatment. So KRAS is emerging now as a very, very important negative predictive marker. And this again sort of gets back to my contention that most of the markers that we use are far better negative predictive markers than positive predictive markers based on the knowledge we have right now. These data are playing out over and over again in a variety of other studies. This is a prospective randomized trial known as the Crystal trial, frontline treatment for patients with metastatic colorectal cancer, randomized to a chemotherapy regimen, FOLFIRI versus chemotherapy plus cetuximab. And again, when the data are analyzed by KRAS status, essentially all of the benefit, here you see the hazard ratio in favor of adding cetuximab in the KRAS wild type patients, but not in the KRAS mutant patients. These data were published just recently in the New England Journal. And this is the forest plot for all of the different variables that were looked at. I won't go through it in detail, but again, you can see KRAS mutant patients, no benefit. KRAS wild type patients essentially have all the benefit from the addition of cetuximab. So these data led ASCO during the year I was president to come out with a new sort of way of updating its guidelines that we called the Provisional Clinical Opinion. We assembled a group of experts who looked at all the data on KRAS and anti-EGFR monoclonal antibodies and basically concluded that the data was strong enough to recommend against the use of these antibodies in patients who had KRAS mutations detected in codons 12 and 13. That was followed, this came out in April last year, as I recall. That was followed in July by FDA actually updating the package labeling for these drugs to, again, recommend against the use of these antibodies in patients with KRAS mutated tumors. Now, it's interesting as this field has evolved that we can begin to drill down even further, beyond KRAS. And this is a slide maybe a little bit difficult to read, but the important thing here is if you look at patients who have KRAS wild-type tumors who would be expected to benefit, there is then a group of patients who have mutant BRAF, which is downstream of KRAS, and those patients also do not benefit from cetuximab treatment. And this was an interesting paper that was published just recently where they began to look at 4 components of the EGFR signaling pathway. So they looked at KRAS, BRAF, loss of PTEN, and mutation in PIK3CA in sort of a convenience sample of 132 patients with metastatic colon cancer. And there's a few interesting things. You can see that in this study, about 20% of patients had KRAS mutations. In the broader experience, it's more like 30 to 35% of patients. About 23% of patients had loss of PTEN. KRAS mutation and BRAF mutation were mutually exclusive. That is, that they were not found in the same specimens from the same patients. And when they began to look at the likelihood of response to cetuximab, essentially they found that all of the responders, that's the blue part of the pie, were in the patients who had no genetic alterations in any of these genes. As soon as one alteration appeared, very few responders were seen, and if there were 2 or more alterations, there were no responders. And they hypothesized, at least from that one could develop an algorithm for EGFR-directed antibody therapy in metastatic colorectal cancer that might look something like that. You evaluate the tumor for its KRAS status first. If it's KRAS mutated, you don't give the drug because it's not going to work. If it's KRAS wild type, then you can evaluate these other genes in the algorithm. And as soon as you find a mutation, you basically kick that patient out of treatment. And ultimately, you might be able to define a sort of a quadruple negative phenotype or genotype where if there are no mutations and normal P10, that the patient has the greatest likelihood of benefit. And when they went back and applied this to the data set that they had, I won't go through it in great detail, but you can see that the 60% of the patients in their data set were the quadruple negatives, and in those patients there was a 50% response rate to single-agent cetuximab. So this sort of subtraction strategy, as I call it, might be a reasonable strategy to pursue to optimize the chances of a patient benefiting from this particular treatment plan. Now, obviously, we have to be concerned about not only what's going on in the tumor, but also what's going on in the germline. And this has been a focus of so much of the work that's going on here in IPIT. And I know many of you are deeply involved in the Tamoxifen story, so I won't spend a lot of time on it, but I just wanted to use it as an important example. Tamoxifen, of course, the most commonly used and arguably the most effective drug in the treatment of breast cancer, is a prodrug. As it comes from the pharmacy, it really has no biological activity. It has to be activated. It's activated primarily by this enzyme, CYP2D6, to 4-hydroxytamoxifen and endoxifen, which are the active metabolites of the drug. CYP2D6, of course, is a polymorphic enzyme in the population, so different people have different levels of enzyme activity. There's a lot of data emerging now, suggesting that the people who have diminished activation of tamoxifen because of genetic polymorphism in CYP2D6 have worse outcomes. So this is just one example of these data. These are people who are extensive metabolizers of tamoxifen. They have a much better relapse-free survival than patients who have diminished activation of tamoxifen. And these data now I think have stimulated an entire field of research. I know a number of you are actively working in this area to figure out, should patients with breast cancer be genotyped if they're candidates for tamoxifen therapy? What do you do if they have a genotype associated with low likelihood of activation of the drug? Is it sufficient to just increase the dose? Will that give them a better therapeutic outcome? I can tell you that there's a big interest at the National Cancer Institute in essentially replacing tamoxifen with endoxifen, the active metabolite, because the view there has been, well, why even worry about the prodrug? Why not just make and give people the active drug? And so they actually have a program ongoing now to synthesize bulk quantities of endoxifen to enable clinical trials with the drug. And of course, the other alternative is many of these women who might be candidates for tamoxifen will also be good candidates for treatment with aromatase inhibitors, which are not subject to this particular kind of variability. And the final important aspect of all this, of course, is that CYP2D6 metabolizes lots of drugs, including other drugs that women take when they're taking tamoxifen, oftentimes drugs that they take to help manage the hot flashes that are a consequence of tamoxifen therapy. So I think oncologists are really beginning to learn now that they have to think carefully about what other medicines they're prescribing to their patients if they're planning to prescribe tamoxifen. And another area where this is important and that Howard knows well, and certainly my colleague Mark Ratain, who I understand received this award last year, knows well, is the whole area of irinotecan pharmacogenetics and the importance of variability in UGT1A1. the enzyme that's responsible for irinotecan glucuronidation. And this is sort of a composite of work that's been done by a number of investigators, but I think pretty clearly shows that those individuals who have the 7-7 genotype and therefore don't glucuronidate the drug well and therefore don't eliminate it well are at very high risk for clinically significant neutropenia as a toxicity of the treatment. There is a commercially available test now that actually can be done to determine the genotype, and in fact, the package label for irinotecan has been changed by the FDA to recommend that patients who have this particular high-risk genotype be treated with a reduced initial dosage of the drug. One of the problems with this whole area, of course, is that we don't know what the reduced initial dosage should be in order to protect patients from toxicity, but still preserve efficacy of the treatment. And so that's a subject of ongoing study to figure out if in fact, one can safely reduce the dose to avoid severe neutropenia, but still preserve the anti-tumor effect. So I want to turn a little bit to discussion about targeted therapy, related to the whole issue of personalizing treatment by understanding how to match therapy to the characteristics of an individual or an individual's tumor has been the whole development of the field of targeted therapy, where we believe that we can identify a particular molecular target in a tumor, develop a drug that hits that target, and hopefully get better treatment outcomes for patients. I think probably the most instructive story here that I'll spend a few minutes on is the whole story of EGFR receptor mutations and the use of small molecule EGFR kinase inhibitors in the treatment of non-small cell lung cancer. So, I think most of you are probably aware that when these drugs first came out, gefitinib was the first one of them. There was a great deal of excitement that these drugs were going to cure lung cancer. And in fact, there were many anecdotes of patients who got drugs like gefitinib and had seemingly remarkable responses to the treatment. Then came a whole series of large prospective randomized clinical trials that enrolled many, many thousands of patients all around the world with the basic study design of chemotherapy plus placebo or chemotherapy plus gefitinib. Every study was negative, and there was a great disappointment and a great lack of insight as to why that might have been the case. Around the time that all those negative results were being reported, came several reports from Harvard identifying sensitizing mutations in the EGFR gene that seemed to be associated with a high likelihood of response to this class of drugs. And we now recognize that there are a number of such sensitizing mutations. They're particularly common in exon 19 and exon 21 of the EGFR gene. And there are also now increasingly being recognized mutations elsewhere in the gene associated with resistance to these drugs, the most prominent being the T790M mutation. And then there's a whole slew of other mutations that have been identified, and their relevance to the utility of these drugs remains to be shown. It's become increasingly clear, though, that EGFR mutation is a very important determinant of whether these drugs work, and in fact, I think whether they should be used. These are data that were published recently, again from Jeff Engelman up at MGH, looking at a composite of 5 clinical trials that explored the use of an EGFR tyrosine kinase inhibitor as part of first-line therapy for patients with metastatic non-small cell lung cancer, where the patient had also had a genetic analysis of the tumor done for either EGFR mutation or KRAS mutation or both. And the remarkable thing about this study, of course, is that this is one of these waterfall plots. The dark blue are the responding patients. They're all patients with sensitizing mutations. You'll notice that it's not exclusively patients with sensitizing mutations. So there must be a little bit more to the story than is revealed just by the sensitizing mutations. Here are the non-responding patients, and these are essentially all patients who either have EGFR wild type or KRAS mutation that makes them relatively resistant to these drugs. And so In the patient population of this study, 38% of the patients had sensitizing mutations, and the response rate to the drugs was 67% in that group of patients. Mind you, if you apply these drugs in an unselected population of patients with non-small cell lung cancer, the response rate is about 10%. So there's a substantial enrichment going on here just by being able to identify this mutation profile. And these patients not only have better responses, but they have a better progression-free survival, as shown here, and they have better overall survival, as shown here. Now, this is a little bit trickier to interpret, and I just want to use this to make an important point about biomarker studies. These are all non-randomized studies. It is clear that the patients who get erlotinib or gefitinib and have an EGFR sensitizing mutation have better outcomes. It's also known that these patients who have mutations have better outcomes even if you don't give them the drugs. Okay, so the EGFR mutation is a prognostic biomarker as well as a predictive biomarker, and I think this is something very important for all of us to keep in mind as we're evaluating biomarkers. If we're going to use a biomarker to identify and select a patient population, it's really essential that we understand whether the biomarker is of prognostic importance as well as of predictive importance, or else we can really be seriously misled by our own data. But this story continues to play out. This is a data from a prospective randomized clinical trial that was done in Asia. So it is enriched phenotypically for patients who are likely to benefit from these drugs. These are Asian patients with adenocarcinoma histology who were never or light smokers. That's the phenotype of patients likely to have EGFR mutation and likely to benefit. But they were randomized prospectively to gefitinib as a single agent or a standard chemotherapy regimen of carboplatin and paclitaxel. And again, if one looks at the overall study results, which are shown here, you can see that over time there's a small and modest benefit that seems to emerge for the gefitinib-treated patients. However, if you begin to break this down by mutation profile, the EGFR positive patients do very well when treated with gefitinib, better than they do when treated with chemotherapy. The EGFR mutation negative patients actually do better when treated with chemotherapy rather than treated with gefitinib. And the data have been supported again by the design of this study, the so-called Saturn Trial. This is a maintenance study, patients with advanced non-small cell lung cancer who get first-line chemotherapy, cisplatin-based, and then if they do not have progressive disease, they were randomized to maintenance therapy with erlotinib versus placebo. And again, if you look at the outcomes of all patients, there's a small incremental benefit in progression-free survival for the erlotinib-treated patients. If you look at this in the patients who have EGFR wild-type tumors, they don't seem to benefit from the erlotinib. If you look at the patients who have EGFR mutated tumors, they seem to have dramatic benefit from the erlotinib. And this is in the setting of a prospective randomized controlled trial. So I think what we are seeing here is the predictive value of the EGFR mutation testing in this group of patients. So these data, some of you will know, of course, that gefitinib, the first-in-class drug here, was actually pulled from the market in both the US and Europe when all those initial prospective randomized trials came out showing that it didn't seem to work. But all of these data, I think, are going to lead to the resurrection of this drug. So in fact, in April this year, the CHMP, which is the European equivalent of ODAC, an advisory committee to advise the government on whether to license drugs, recommended the approval of gefitinib or Iressa for adults with locally advanced or metastatic non-small cell lung cancer who had activating mutations in the EGFR receptor gene. And then in July, the manufacturer of this drug, AstraZeneca, announced that, in fact, the European Commission had granted marketing approval for this drug in patients who could be identified by the biomarker of EGFR mutation status. So here's a drug that has had a complete life cycle from coming on the market as a single agent, lots of excitement, great disappointment when randomized trials turned out to be negative, pulled off the market, new insights into biology, new trials and biomarker selected patient populations that then have resulted in the drug now coming back on the market, at least in Europe and potentially in the US as well. So I want to say a few words that maybe have more to do with policy than research. We have lots of drugs in development for cancer right now. The estimates are all over the board, but there probably are at least 800 drugs in development for cancer. We have a very low success rate. So the estimates are that if one looks at the success of getting a drug from phase 1 to marketing, we're only hitting about 5 to 8% of the time for cancer drugs. Clearly, we need to do better than that. We have way too many drugs in development. We have way too much unmet medical need. We can't afford to be missing 90+% of the time. It's way too expensive. So why the high failure rate? Well, I think this is still the fundamental problem. Even though I started this talk saying we know more about cancer biology than we ever have, we still don't know nearly enough. And every time we learn something, it reveals a new layer of complexity that we may not have appreciated previously. So we need to learn more. We have very poorly predictive animal models in most cases, and I know that some of you are working to make better animal models, but most of the animal models that we've used over the years are not very good at predicting what's going to work in people. We have cured millions of mice with cancer, but we have cured far fewer people. We have not done particularly insightful phase 2 trials, the trials that are absolutely essential in informing the decision as to whether to launch a pivotal phase 3 trial. So we need to do a better job there. A huge problem, of course, that's central to this whole area is that we deal with very heterogeneous patient populations, and I would contend that we still have inadequate biomarkers to enable enrichment, particularly biomarkers that are likely to identify patients who are going to benefit from the treatment. And then we have some lack of agreement between the investigator community, the pharmaceutical industry, and the regulatory authorities about what constitutes clinical benefit to a patient. And of course, that is a critical element of the FDA approval process, is that the drug sponsor has to demonstrate, quote, clinical benefit before the drug can be approved for marketing. So how can we do it better? Well, I think for the time being, it may well be that if we get a biospecimen on every patient, it will allow us to at least generate the knowledge that we need to have to then develop drugs more efficiently. I'm still not convinced that the future in terms of oncology practice is going to necessarily rely on doing molecular profiling on every single patient's tumor on every single time that a treatment decision needs to be made. That still seems to me to be relatively impractical. I would hope that as time goes by, we'll be able to actually substitute sophisticated functional imaging for a lot of what we're doing now with biomarker work, but time will tell. If we're going to use biomarkers, we need to use analytically validated biomarkers that are run in a CLIA-certified laboratory if they're going to be used for clinical decision making. We need a more flexible regulatory system that will allow us to develop drugs and biomarkers together in a much more efficient fashion. I'll come back to that point in a minute. We need reimbursement and regulatory incentives for development of diagnostics companies. The companies that make these tests will tell you that it's very hard to make ends meet because diagnostic tests are not reimbursed nearly at the rate at which drugs are reimbursed, even though the diagnostic test can actually be of critical importance in determining an optimal treatment plan for a patient. And then I think importantly, and we're seeing some movement in this direction, we need the pharmaceutical industry to be willing to trade off market share for treatment duration. So what do I mean by that? The days of the blockbuster chemotherapy drug, I think, are coming to an end. The day when a drug can be developed that every patient with a particular diagnosis gets that drug, come hell or high water. I think those days are pretty much over. So instead of giving every patient a treatment for 3 or 4 months, because most of them are ultimately going to have progressive disease, the goal for the pharmaceutical industry should be create a treatment that will work in a segment of the population who can stay on the treatment for a long period of time, and they'll generate their revenues in that way. Think about how Gleevec a drug for a series of very uncommon diseases is a billion-dollar-a-year seller for Novartis. When the stars all line up, when we have real insights into the biology and we have really good drugs, we can make progress rapidly. And this is the data on the phase 1 trial of the Genentech hedgehog inhibitor that appeared recently in the New England Journal of Medicine. I've been doing phase 1 trials for 25 years. I've never yet reported one in the New England Journal of Medicine. But this is a good one, and it shows you that basal cell carcinomas of the skin, the most common cancer in the world, but almost always treatable with local excision. Many of them are driven by mutations in the hedgehog pathway. I don't have time to go through it in detail, but the point is that when the mutation is a driver mutation in the tumor and you give a drug that interrupts that pathway, you can get lots of anti-tumor responses. And all of the patients here shown in light or dark blue are complete or partial remissions on a phase 1 study, and many of them for fairly long duration. Similarly, we've gotten a lot of insights recently into melanoma biology and the importance of BRAF mutation in melanoma. And it's known now that activating mutations in BRAF are found in 50 to 60% of patients with melanoma, and 90% of those mutations are the V600E substitution. This is a new drug presented at ASCO this year in a phase 1 trial. 9 of 15 patients who had the V600E mutation responded to this drug at doses of greater than 240 milligrams given twice a day. Median progression-free survival was 6+ months, with toxicity that most of us would consider to be acceptable and manageable. So again, exceeding a 50% response rate in a refractory malignancy like melanoma in a phase 1 trial, I think is truly a remarkable progress. What are our challenges going ahead? Well, every time we think oncology is going to get simpler, it gets more complicated. To some extent, of course, we have many more drugs than we ever used to have, and we need to understand how to better use those drugs. We also have many more diseases than we used to have. We used to have non-small cell lung cancer. Well, now we have adenocarcinoma of the lung. We have squamous carcinoma of the lung. Different chemotherapy regimens seem to work better in those different histologies. Now we have EGFR mutated non-small cell and EGFR wild type non-small cell, and we need different therapeutic approaches in those patients. In colon cancer, we have KRAS mutated, KRAS wild type, and undoubtedly, there'll be more and more subpopulations of all of these tumor types that are recognized as time goes by. Because of the nature of the drugs that we're developing now, we're using more placebo-controlled trials. That makes patient recruitment more complicated. We're also using more randomized screening trials as opposed to the old-fashioned single-arm phase 2 trial. That requires larger numbers of patients. Because we're using more progression endpoints than response endpoints, it takes a longer time to reach those endpoints, and it requires more expensive documentation. So everything is actually more difficult in cancer drug development than it used to be in terms of the operations of the trials. The hope is that by better matching the therapy to the molecular characteristics of the patients, we'll actually get more effective treatments. We'll have a higher probability of demonstrating efficacy, and although it may take a more complex trial design to show that, we'll be able to show it more frequently. And we also have a lot more regulatory complexity than we used to. So our opportunity, of course, is that we can use biomarkers to select patients who are either likely or unlikely to benefit from treatment, and use those biomarker strategies to enrich the patient population. That hopefully would give us a larger effect size in a clinical trial. That would enable us to do the trial with a smaller sample size and therefore get more rapid results and, of course, get greater benefits to patients more quickly. The risk in all this, as I see it, is, again, comes down to biology. If we don't have an adequate understanding of the target, then we're in trouble, because we may be trying to develop a drug against a target that is biologically unimportant. We also have some risk associated with choosing the biomarker incorrectly to enable enrichment. Take the example of using EGFR expression to enrich patients who are likely to benefit from cetuximab. When cetuximab came on the market, it was labeled for use in patients with EGFR overexpressing colon tumors. Well, it turns out that EGFR expression is fairly irrelevant to whether or not cetuximab works, so it was the wrong biomarker. Or we may have circumstances where we have the right biomarker, but we have a suboptimal technology to assess it. And here I give you the example of using immunohistochemistry to assess HER2 overexpression as a way of predicting who should get trastuzumab or Herceptin therapy. That was the original biomarker test, but subsequently we learned that using FISH to look at HER2 amplification is probably a better test. Using these tests, we need to screen more patients. If the biomarker has a low prevalence in the population, we may have to screen 10 patients to find one who can actually be eligible to go on a study. And we have to deal with these issues of time to study, activation, and accrual. So I've given you my hypothesis that it's difficult to find positive predictive markers. Why is that? And I think one answer to it, at least, is that, frankly, there are many ways to fail. Cancer continues to be very wily in developing means of escaping the effects of any kind of therapy we throw at it. So we've learned now that you can escape from a targeted therapy by having mutation at the binding site so that the drug no longer really hits the target. You can have downstream mutation or downstream activation of the pathway that you're intending to inhibit, so even if you inhibit the target, the pathway is still active. You might have feedback upregulation of the pathway. You might have activation of a parallel pathway that essentially circumvents the block. Or you might have some sort of pharmacologic resistance, and we shouldn't forget that we're still giving drugs to people, and targeted therapies are still drugs that we have to optimize in terms of their pharmacokinetics and pharmacodynamics. And again, I bring you back to the example of the EGFR pathway. Here's the pathway. The receptor can homodimerize. The receptor can heterodimerize. There can be an alternative receptor that's activated that signals down the same pathway. The pathway can signal down at least two different arms and can be blocked at various levels. And if you, again, look at this cartoon, so here's the optimal situation where the pathway is active, you apply the drug produces a block in the receptor signaling, and the patient responds. But if you have a resistance mutation, in this case, say the T790M mutation, the drug doesn't bind properly, you don't inhibit the pathway. Or you might have a circumstance where you block the EGFR pathway, but the tumor amplifies the MET pathway as an alternative way of signaling down this pathway, and then it circumvents the block. Or you may have some sort of downstream activation where you block upstream, but you get a PI3 kinase mutation downstream that activates the pathway. Just going to skip ahead in the interest of time. So I just wanted to close with a few comments about some of the regulatory challenges that we face in developing personalized medicine. And I put them in three categories: co-development of drugs and molecular diagnostics, development of combination targeted therapies, and then this whole issue of this rapid proliferation of laboratory-developed tests, which are being directly marketed to physicians and consumers without really a good evidentiary basis to their utility. Now, with respect to the issue of drug and biomarker co-development, so I had an opportunity to participate in a panel that presented its results at a meeting hosted by the Brookings Institution in September. And the charge to our panel, as shown here, was to facilitate the accelerated development and approval for a cancer therapy used in a population defined by a specific diagnostic test. So this is a very interesting discussion because I was sort of the naive kid on the block, because the other discussants were two former FDA commissioners, Mark McClellan and David Kessler, and David Epstein, the president of Novartis Oncology, and David Zadransky from Johns Hopkins, who's an expert in cancer biology and tumor markers. And it was a very interesting little discussion group to be in. But we came up with this proposed policy for what we call targeted approval. So here are the criteria. The drug must be indicated for cancer. The diagnostic assay must be analytically valid. The drug must demonstrate in a population defined by the assay a pre-specified statistically significant change in an endpoint that's reasonably likely to predict clinical benefit. So if all those criteria are met, then we would propose that the FDA should approve the drug for its intended use in the subpopulation defined by the test, that CDRH, the Diagnostics Division of FDA, would approve the device for the claim of identifying patients who were studied in a drug trial, that is, patients who are likely to benefit from the treatment, with the caveat that the test has not been shown to be useful in identifying patients with an expected lack of effect, and that post-marketing studies would then need to be done of both the drug and the test to prove their ultimate utility in producing clinical benefit for patients. And while those post-marketing studies were ongoing, that CMS and other payers would not reimburse for off-label use of the drugs until the post-marketing studies were concluded, unless it was in this unique context of what Medicare or CMS calls coverage with evidence development. We also had another panel at the Brookings Institution on combining new molecular entities, and the concept here being one that I think everybody is thinking a lot about. How do you take two targeted drugs that by themselves have minimal anti-tumor activity and put them together to make a blockbuster regimen, and how do you develop that combination in a way that is scientifically valid and meets regulatory standards? And that's really the challenge. And we came up with a number of paradigms for how to do this, and I don't have time to go into it in detail, but if you're interested, there's a white paper on this on the Brookings Institution website that you can look at. And basically the proposal was to do a phase 1 trial in sort of a ladder design, looking at combining the two drugs to come up with one or more recommended phase 2 doses for the combination, then doing a randomized phase 2 study comparing drug A plus drug B versus some standard of care to try to get a signal as to whether or not the combination looked more promising than a standard of care, and then moving that directly into a phase 3 trial. This would be a paradigm that would be used where drugs A and B have minimal anti-tumor activity in their own right, and there really would be very little justification for using them as single agents. The other possibility, of course, is that the drugs do have some single agent activity, but you would expect that the combination would be far more effective than either drug used by itself. And again, there were sort of a paradigm that this panel came up with that you can read about on the white paper for how to proceed with drug development under these circumstances. Finally, just one quick word about the regulation of diagnostics in this country. I think some of you will realize this, but you should all recognize that the way in which diagnostic tests are regulated is in two different pathways. So the Clinical Laboratory Improvement Act, or CLIA, specifies the laboratory practices that have to be followed by clinical labs. And a CLIA-certified lab has to establish performance specifications for laboratory-developed tests. It is not required under CLIA to demonstrate the clinical utility of any test that is developed by a laboratory, only to demonstrate that the test is done in an analytically valid way according to CLIA SOPs. The alternative pathway is to get FDA approval of a test, and this is typically in the context of approving a test kit that is used to perform a diagnostic test for an intended use. And there are several different kinds of FDA approval that can be sought or obtained based upon the risk associated with the use of the test. What FDA looks for in their review is analytical validity of the test, the clinical validity of the test, and the proposed labeling by the sponsor. And so here's a couple of examples. These are FDA approved tests. The Veridex circulating tumor cell test, which is approved for enumeration of circulating epithelial cells. It doesn't tell you whether enumerating them is actually good for anything. It just tells you that the test is FDA approved to use for that purpose. Or the Hercept test, which is the immunohistochemistry test, the DACO test for guiding Herceptin therapy. Those are FDA approved tests. These are not FDA approved tests. These are tests done under CLIA. The Oncotype DX test that I mentioned previously, not an FDA approved test, but performed under CLIA for assessment of risk of recurrence of node-negative ER-positive breast cancer. And KRAS mutation testing or EGFR mutation testing are also examples of laboratory-developed tests that are done under CLIA. Here's the problem. We have all these tests. We have existing regulatory paths that do not require demonstration of clinical utility. We have lots of direct-to-consumer advertising, and that results in lots of physician confusion and increasing physician responsibility. We are seeing patients who come into us with a printout of some test results from their buccal swab or some test results from their tumor block and say, "Doc, here's my profile. Tell me what treatment I should take." And more often than not, it doesn't help us in guiding their treatment. So I think we need reform at three levels. We need policy reform to clarify the roles of CMS, which administers CLIA, and FDA with respect to test approval, labeling, and reimbursement. We need regulatory reform whereby FDA should issue guidance to determine the level of evidence required to support claims of clinical utility for tests. And then, of course, we need practice reform. We need guidance to physicians on how to interpret and use these in vitro diagnostic tests. And I think if we accomplish all these things, we will get to the vision of personalized cancer care, which I would suggest derives from understanding the germline, understanding the tumor, and delivering care with a personal touch to our patients. So thank you very much for your attention.